Hey everybody, picking up here with chapter 17, digestive system, slide 45. I believe that's where I left off last time. And I apologize, I'm actually making this video during my office hours, so I might periodically check just to make sure that no one popped in. Normally it beeps at me when someone comes in, but I might just check. Okay. So, the pancreas, this is where we're starting off. The pancreas is an endocrine gland and an exocrine gland. So we mentioned the endocrine functions back in chapter 13, right? Insulin, glucagon, somatostatin. It doesn't mention somatostatin right here, but we did talk about that a little bit. The pancreas, though, is also an exocrine gland. It makes digestive enzymes as well as, bicarbonate that are going to go and empty into the duodenum of the small intestine through, it travels there through what we call the pancreatic duct. So this combination of digestive enzymes and bicarbonate and water, we call this the pancreatic juice. You can see where the pancreas is located here. We've kind of been there, done that, but we'll look at the structure just a little bit more again. So the pancreatic acinar cells are going to make up most of the pancreas. You probably noticed that when you looked at the pancreas under the microscope in lab, that it was a little bit harder to find the islets of Langerhans, the endocrine cells, or the pockets of endocrine cells, than it was to find the acinar cells. The acinar cells are everywhere, right? So tons and tons of exocrine acinar cells making those digestive enzymes. The pancreatic duct is going to carry the digestive enzymes and the bicarbonate and the water all the way to what we call the ampulla of Vater. So you see that. Let me actually get my highlighter out here. This ampulla of Vater, otherwise known as the hepatopancreatic ampulla. You can use either of those terms. You see that the pancreatic duct ends up there along with the common bile duct. So we kind of we talked about the common bile duct a little bit, but we'll talk about it more in this chapter. But the common bile duct empties there as well, and then the ampulla of Vater empties into the duodenum of the small intestine, right? And so pancreatic duct, along with the common bile duct from the liver and gallbladder, empties into the duodenum of the small intestine. Hepatopancreatic sphincter, or as I like to call it, the sphincter of Oddi, because we love to name things 12 times in biology, right? That sphincter of Oddi is going to be, right there at that ampulla of Vater. And it, when it's relaxed, when that sphincter is relaxed, the substances are able to flow out of the ampulla of Vater into the duodenum. But when it's contracted, then that opening is pinched off, and so nothing can flow into the duodenum from those ducts. Let's see a picture of this, and I'm very, very exceedingly particular. You probably noticed that already. I'm very particular, but very particular about these bile duct names. So you'll see here the common bile duct right here, and you'll also see the pancreatic duct right here. So make sure that you get the names correct. This pancreatic duct comes over here, and this common bile duct it actually starts right here. So this, I know we haven't really gotten into all the details yet, but this duct right here, that's the cystic duct. So when the gallbladder contracts, bile moves into the cystic duct and then into the common bile duct. So the common bile duct is shown there, and I put it in red. And the common bile duct is going to come down here and go the same ampulla that pancreatic duct empties at. So they're going to go there right there together, okay? And let me zoom in here. Let me take these things off of this picture. Zoom in. Insert. Xbox. There we go. It should let me zoom now. Excellent. Okay. So you can see that common bile duct and the pancreatic duct kind of merge together at that ampulla, but you've got that sphincter that kind of controls the flow. And actually, the sphincter really controls flow into the ampulla, so it's kind of like right kind of behind the ampulla. And then from the ampulla of Vater, you have the ability to move those substances into the duodenum. Okay. All right. So looking at everything that's in that pancreatic juice, you can see all these things listed here. So there are tons of things in this pancreatic juice, right? So I mentioned that there are enzymes here, right? Well, there are a lot of enzymes here. We have got pancreatic amylase. Now, we have talked about amylase before, not in the pancreatic sense, but it does the same thing. So we know that we have amylase in our saliva too, but we also have the pancreas making it, and it does the same thing. Like I said, it breaks down your polysaccharides, right? So you see here, breaks down starch and glycogen. Well, starch and glycogen are polysaccharides, so it's going to break them down into simpler sugars, these disaccharides, which are like two single sugars stuck together. That was kind of hard to say. Two single sugars stuck together. There we go. Pancreatic lipase. So as the name implies, lipase breaks down lipids, right? So big example of that are your triglycerides. So triglycerides, you break those down into 3 fatty acids and 1 glycerol molecule. Okay, let me just see. Oh, nobody's here. By the way, people really haven't been coming. And so that's why I thought I could make this during office hours. So don't forget about office hours, okay? You can always come to my office hours, my virtual office hours. Okay, trypsin. So trypsin, understand trypsin is actually secreted as trypsinogen. So it gets secreted in an inactive form, and it has to get converted into trypsin. And interestingly, it doesn't get converted into trypsin until after the trypsinogen makes it to the small intestine. So understand this. I know it's kind of a strange sequence of events, but you've got the pancreas making trypsinogen, and then trypsinogen travels to the small intestine and gets converted into trypsin by a chemical called enterokinase. So, so trypsin, I don't want you to think that trypsin is an enzyme of the small intestine. It's still made by the pancreas. It just doesn't get, activated. Trypsinogen doesn't get activated into active trypsin until that destination of the duodenum has been reached. Okay, so what does trypsin do? Well, trypsin does actually a couple of things. It's kind of a busy enzyme. So one thing it does is it breaks down proteins into smaller peptides. And it's also going to activate other enzymes. So it's a digester and an activator. So trypsin is going to be responsible for getting these guys made. So you see here, at least getting activated, not made, getting activated. So chymotrypsin is actually secreted as chymotrypsinogen. And carboxypeptidase is actually secreted as procarboxypeptidase. So those two enzymes are not active. Chymotrypsinogen and procarboxypeptidase are not active. And they, along with trypsinogen, go to the small intestine. So trypsinogen, chymotrypsinogen, procarboxypeptidase, they all travel to the small intestine together. When they get to the duodenum, enterokinase converts trypsinogen into trypsin. And then trypsin turns around and converts chymotrypsinogen into chymotrypsin and converts procarboxypeptidase into carboxypeptidase. So it kind of activates its neighbors there. And what do chymotrypsin and carboxypeptidase do? Well, chymotrypsin kind of does the same thing that trypsin does here, at least in terms of the digestion, right? So it's going to break down proteins into smaller peptides. Carboxypeptidase, it's a similar function. It's still in the protein catabolism pathway, but we're working on getting things a little bit smaller. So carboxypeptidase is going to break down peptides into amino acids. Understand though, like I said, those are all part of protein catabolism. So if I were to, let me just put this new little slide here. I'll do a blank slide. I'm going to draw a little bit on here. So proteins, proteins we know are polypeptides that are folded in a specific way, right? So these proteins are made up of amino acids at their most basic level, but understand, so let me put here, folded polypeptides. Okay, folded polypeptides. And then we break proteins down, we can get them into smaller polypeptides and peptides. So, peptides, you've got, you still have multiple amino acids joined together, just might not be long as a, as a polypeptide. So polypeptide means that there are kind of a lot of amino acids there, right? And then from there, we get broken down into amino acids. So we kind of have to go in the sequence. So the things that are working, that have been working on proteins in their protein state are going to be, we talked about pepsin working on proteins back in the stomach, but here in the small intestine, we have trypsin and chymotrypsin working on proteins, again, getting them into, down to that peptide stage. And then carboxypeptidase, breaking those peptides down into amino acids. So as this name kind of implies here, carboxypeptidase, you see peptid, that might help you remember that we are working specifically on peptides. So you use that as a little clue. Nucleases, as the name implies, we are breaking down nucleic acids. And then bicarbonate. Bicarbonate is not an enzyme. Bicarbonate's not breaking anything down. It's just neutralizing acid, though, right? So this is going to make things more alkaline, buffer stomach acid. So again, think about where that pancreatic duct goes. So let me put this picture back up here. Where does the pancreatic duct go? The pancreatic duct goes to the duodenum. Well, what else went to the duodenum? So what's coming in this way? Chyme from the stomach, right? Chyme from the stomach. Chyme from the stomach should have what kind of pH? Should be very low pH, very acidic, because all that hydrochloric acid, right? So we have all of this acidic stuff coming into the duodenum. We need that bicarbonate, right? We need that bicarbonate bad to try to neutralize that acid, because we don't want all that acidity in our small intestine. Okay. So here, this is a good picture. I'm gonna kind of go fast through this picture and go to the next one. So zymogen, by the way, is just the term that we give to that kind of package of inactivated enzymes. So your proteolytic pancreatic enzymes, trypsin, chymotrypsin, and carboxypeptidase are actually in an inactive form, right? We know that trypsin is actually trypsinogen, chymotrypsin is chymotrypsinogen, carboxypeptidase is procarboxypeptidase. So that's how they come initially in that inactivated form in a little package that we call a zymogen granule. And those zymogens go from the pancreatic duct into the duodenum of the small intestine. Now I'm going to kind of go to this slide. I like this slide a lot. This slide gives you a lot of good info. So we have substances, coming from the pancreatic duct. So we've got among those substances being your digestive enzymes, and those proteases are going to be in their inactive form. And actually, if they were activated too early, they could damage the pancreas, by the way. So that's why they're not active until they get to the small intestine. And it kind of makes sense. I mean, what on earth do we need to be digesting in the pancreas anyway? We don't have food going there. So, it makes perfect sense to not really activate these until we get to this point. But you can see trypsinogen right here. So trypsinogen is going to interact with enterokinase in the duodenum of the small intestine. And enterokinase converts trypsinogen into trypsin, which is active. And trypsin, remember, will go down and work on, or it's going to break down proteins, sorry. It's going to break down proteins there in the small intestine. But it's also going to activate two other enzymes, right? So it's going to convert chymotrypsinogen into chymotrypsin, and it's going to convert procarboxypeptidase, sorry, I got tongue-tied, into carboxypeptidase. And so those conversions there are mediated by trypsin. So I really like to ask these types of questions. So there are, you can expect enzyme questions on the exam, and among those questions could be, okay, what activates this? What activates that? We do have several things getting activated, as you can see, because we saw it in the stomach, and we see more activation steps here. Not all enzymes are secreted in their active form. Okay, so regulation of pancreatic secretion. So we actually talked about some of this before in a previous slide. Oops. So you look at this image, look first at number 1. It says acidic chyme enters the duodenum. So we got these acidic stomach contents coming into the duodenum of the small intestine. And that's like a bell going off, right? We need to neutralize that acid. And so the intestinal mucosa releases secretin. Secretin is going to go through the blood to the pancreas, and secretin stimulates the pancreas to secrete bicarbonate. And bicarbonate goes into the pancreatic juice, and then that pancreatic juice containing lots of bicarbonate in it is going to head into the duodenum of the small intestine to neutralize the acid in those recently arrived contents there from the stomach. So I know I mentioned this in the previous video, but just don't get secretin and CCK confused, and make sure you're always specific. So again, you can't tell me secretin stimulates the pancreas. You can't say secretin stimulates the pancreas to release pancreatic juice. What do I need to know about secretin? It stimulates the pancreas to release bicarbonate ions into the pancreatic juice. Now, would CCK be secreted at the same time? You bet it would. You bet it would, because if you have stuff moving from the stomach into the duodenum, you need both CCK and secretin out there. But they, CCK and secretin do different things to the pancreas. So make sure that you're very specific when you're talking about those in terms of what exactly are they making the pancreas do. Okay. So, and here we go. Speak of the devil. So secretin stimulates pancreas to release pancreatic juice high in bicarbonate ions. CCK stimulates pancreas to release pancreatic juice high in enzymes. So if you have them both being secreted at the same time, which you would, then you would have pancreatic juice that is rich in both of those things, okay? But, both bicarbonate and digestive enzymes. So just make sure, though, that you're specific, because I'm going to ask about those functions for sure. Okay. Still nobody there. Okay. Alrighty, and you can see that talk about CCK a lot. So you can guess it would probably honestly be very unlikely that I won't ask you about CCK on the exam. So it's something that I usually ask about, and because it's multifunctional, sometimes people forget all those three functions. So just make sure that you study that really, really well. So again, the three functions with CCK are not all shown on this picture. This picture actually only kind of vaguely talks about two, but look, CCK secretion, you see a plus there going to the gallbladder, because CCK secretion triggers the gallbladder to contract and release bile into the cystic duct and beyond, right? So that we get bile then delivered from the common bile duct into the duodenum of the small intestine. And CCK, you see this plus by the pancreas, but again, be specific. What is it causing the pancreas to do? It stimulates the pancreas to release digestive enzymes. All right, so CCK stimulates digestive enzyme secretion from the pancreas to causes the pancreas to release digestive enzymes into that pancreatic juice. And the third thing that's not on here is what CCK is doing to the stomach. So what does CCK do to the stomach? CCK decreases stomach activity, okay? So, CCK stimulates two things and inhibits something else. So sometimes people get that confused too. So CCK tells the stomach to stop being so active, okay? Tone it down, stomach. That's CCK. And then CCK, remember what causes it to be secreted. CCK is secreted when stuff moves from the stomach to the duodenum, okay? So by stuff, I mean chyme, right? So you have obviously a need to digest things that move into the small intestine from the stomach. And among those things in the chyme, there could be fats. And if there are fats, then we need bile. Okay, so it makes sense why the gallbladder is going to contract. And if there are nucleic acids and proteins and fats, we also need, the digestive enzymes coming from the pancreas. Why? Well, go back at the pancreatic enzymes. We have pancreatic enzymes that are going to break down fats. We have pancreatic enzymes that are going to break down proteins. We have pancreatic enzymes that are going to break down nucleic acids. And I forgot about carbohydrates too. If there are carbohydrates in there, we've got to have that pancreatic amylase, right? So we need all of those enzymes to work on that stuff in the small intestine. So you need that. And you need the stomach to kind of tone it down a little bit. We don't want too much going into the small intestine at one time, but also we are moving on from the stomach. We've done what we needed to do in the stomach, and now we have to digest things in the small intestine. Okay, liver. So the liver is the largest internal organ. Big, very big. I know sometimes, I know we're not doing dissections in my class this semester, but a lot of times people are surprised. You open up the abdomen and it's just like enormous liver. Like, wow, is that really one whole organ? Yes, one whole organ. It has different lobes, but it's It's a very big organ. It's located in the upper right and upper left quadrants. The bulk of it's going to be in the upper right just because of that enormous right lobe of the liver. And it's, yeah, just inferior to the diaphragm, reddish brown. I'm honestly not going to ask you what color the liver is. It doesn't seem like that's the most important thing, you know, not the most important take-home message here. Well supplied with blood vessels. That's something to note because the liver, honestly, it has a goo gob of function. So it does a lot of things. It makes a lot of things. It's very metabolically active. We need a lot of oxygen. It has a high oxygen demand because it's doing a lot of chemical reactions, needs a lot of energy. And then because it's doing a lot of cellular metabolism, it's also going to, or cellular respiration, it's going to be producing a lot of CO2. So when you do those cellular respiration reactions, you make CO2. So there's a lot of CO2 to take away as well. And we also have detoxification occurring in the liver, destruction of red blood cells occurring in the liver. So, I mean, it's hard to do those things if you don't have a lot of incoming blood, right? So it's definitely going to be rich with blood vessels here. Okay, so this is something I could ask you in a picture. I could ask you to identify the lobes of the liver, and you see in this image, or these images, you can see them labeled. So this is just a front view. This is looking at an anterior view, and you can see the huge right lobe and the little left lobe, that little tiny left lobe off to the side, right? This picture to the side is what it would look like if we took it and flipped it up. So we took, you know, if we took, you know, this end here and flipped it up, okay? So that's off to the side. Hold on. And you can see when you look at it from the underside, you can see the quadrate lobe and the caudate lobe. So the quadrate lobe is by the gallbladder. You can see it's kind of between the gallbladder and the left lobe of the liver. And then the caudate lobe is between the inferior vena cava and the left lobe of the liver. So use the gallbladder and inferior vena cava as good models. Not models, I'm sorry. Landmarks is what I meant to say on your models and diagrams. So good landmarks, letting you know where those lobes are. The only trick I can tell you for those two, because normally when people get confused, it's with those last two that I mentioned, the quadrate and the caudate lobe. Those are the ones that get turned around. They usually get the right and left, good handle on that. But the quadrate and caudate give them trouble. I just use a little bit of letter association. I know that's kind of silly, but sometimes it works, and you know, do what works, right? So inferior vena cava. So I just think of cava and caudate both start with CA, and it helps me remember that that caudate lobe is down there. You know, you can't use letter association with quadrate. There aren't too many things around here to start with the letter Q. But you know, if you remember where the caudate is, then you know that the quadrate is, well, it's the other one, right? So it's the other little lobe. And quadrate, again, it's by the gallbladder. Okay. This is another view from the backside or the underside of the liver. It's just, instead of being flipped up, it's just simply turned around. So I put both in here. When I first made this PowerPoint, I noticed that some students kind of, I don't know, they got kind of confused as to this picture B over here, and so I found this image as well. So, you know, if you're not sure which one you're looking at, again, just look for your inferior vena cava and your gallbladder, and those will be your landmarks. That should help you. Okay. So let's talk a little bit about the hepatic portal system and the liver structure. So you see this kind of hexagonal structure here. That's called a hepatic lobule. Hepatic lobule. So a hepatic lobule, let me just outline it. I'll outline it in yellow. They make it look so hexagonal here. It's like I'm in geometry. So you've got a hepatic lobule, and you can see some more, you know, over here. They just get kind of cut off in the image, but you get the idea. Now, if you were to look at the center of that lobule, should switch colors, but that's okay. That's the central vein right there. Okay, so this is a blood vessel, and that central vein takes blood out of that hepatic lobule. Okay, so blood that's going through there is going on its way out. Well, how does blood get in to hepatic lobules? Well, you see these things over here to the side. These are called your hepatic portal triads. And you can see them just around all the corners. I'm not going to circle all of them, but you get the idea. Hepatic portal triads. And there are 2 blood vessels and 1 bile duct at each of those hepatic portal triads. So as the name implies, triad means that there are 3 things there. And the 3 things are a bile duct and a branch of the hepatic artery, which you could also call a hepatic arteriole. And there is a branch of the hepatic vein, hepatic portal vein, which you could call a portal venule. I almost skipped the word portal. Please don't skip the word portal. Okay, so this blue vessel right here. This is not a branch of the hepatic vein. This is a branch of the hepatic portal vein. Okay, it's very, very important that you understand that because the hepatic vein leaves the entire liver. The hepatic portal vein brings blood into the liver. So they're going in opposite ways, okay, from this organ. One's coming into the organ and one's leaving it. So they're very, very different. So you have blood coming into this hepatic lobule through each of these 2 vessels here. So blood comes into the liver through hepatic artery and through the hepatic portal vein, and then those blood vessels branch off into these arterioles and venules, right? So you have the hepatic arteriole, which means a branch of the hepatic artery, bringing in oxygen-rich blood. Liver's going to need a lot of oxygen, right? Going to need a lot of oxygen, definitely so. And then you have this hepatic portal venule, aka a branch of the hepatic portal vein, bringing in oxygen-poor blood that has actually already been through capillaries in the stomach and small intestine, large intestine, spleen, and pancreas. So it's already been to all of your digestive organs and your spleen too, and done exchange in those organs. But then it's come into this vein, the hepatic portal vein, and branched off into these hepatic portal venules and is going into each hepatic lobule to end up in yet another set of capillaries. So we call this type of system a hepatic portal system because we're going through 2 sets of capillaries. And so where you see here that the hepatic arteriole and hepatic portal venules come together, that little spot we call hepatic sinusoids, and those sinusoids are capillaries. They are capillaries, and you're doing exchange again. So things are going out of the blood and into the cells, and things are coming from the cells and going into the blood. So we're doing movement of substances back and forth in and out of those hepatic sinusoids. And at the end of this exchange process, blood will then go into this central vein and leave that hepatic lobule. And then those central veins come together to form the hepatic vein, which exits the liver. So that is the flow of blood through the liver. Now, I did mention that in the hepatic portal triad, we also had one additional component, and that was the bile duct. And here I'm okay with you just using that generic term bile duct. I don't really know that there's a specific, any more of a specific name for it right here than that. So that green guy right there is a bile duct, and the bile is moving this way. Why? Because the hepatocytes they're making the bile, right? So, all these cells out here, these are all called hepatocytes. And they are all making bile, and the bile is going through a series of canals called the bile canaliculi. And then those canaliculi will end up sending their bile to a bile duct, and the bile leaves the hepatic lobule this way. And where do those bile ducts right there end up? Well, they end up going into what we call the right and left hepatic ducts. And so we'll look at the additional ducts coming up soon, I think. Yeah, I'll look at them down there by the gallbladder. I know I have some pictures. I definitely want you to understand bile flow. And I could go through it right here, but it seems like it would make so much more sense to y'all if I had the picture up at the same time. So let me just kind of hold on to that. We'll tuck that in our pockets and come back to it in a minute when we have a nice image to go along with it. Okay, I keep moving my, there we go. That's good. Okay, moving on to here. Oh, look, another picture of the same thing. The only other thing here that wasn't in the previous slide is the Kupfer cell. So you see up there where it says, oops, where it says Kupfer cell at the top, Kupffer, those are phagocytic cells kind of swimming around in those blood vessels there, you know, those hepatic sinusoids in the liver. Their job is phagocytosis, like I said, they're phagocytic. So they're going to be engulfing things. Remember, we do have detoxification and destruction of red blood cells and things like that occurring here in the in the liver, right? So definitely need some phagocytosis going on. So Kupfer cells. Do you know that specific name? There's some picture, I can't remember where it is, but I remember there's some picture somewhere, might be in the lab book, that just calls it like a reticuloendothelial cell. That's really not specific enough, so make sure that you call it a Kupfer cell. Oh yeah, okay, I'm seeing all this stuff at the bottom. Did I say all that? Yes, I said all of that. Okay. Okay, liver functions. And yes, you do need to know them all. There are a lot. The liver does so many things. So many things. So think about if you have liver dysfunction, how dramatic that can be because it affects so many things. So let's talk about these. Some of these you should already know, in all honesty. So like these two at the top, we totally have been there, done that in chapter 13, right? So we know that the liver produces glycogen from glucose. Glycogen is a storage form of glucose. This is going to be happening if you have insulin secretion. And then we also know that we can go back the other way and break down glycogen to make glucose. That would be going on if you have glucagon secretion, right? So we've done that. And then converts non-carbohydrates to glucose. That's gluconeogenesis. We know that glucagon and cortisol cause that to happen. So we have again been there. Now the somewhere here. Oh, we did talk about plasma proteins. We talked about how a lot of those are made by the liver, so that should be review. I don't know that it's on here. But I mean, well, it does say, you know, it says plasma proteins, but you do have a lot of clotting factors made by the liver as well. So, you know, you can kind of think about that as well. I mean, you are going to have clotting factors moving through the blood. So, I mean, you could have that kind of be included here in this synthesizes plasma proteins bullet point. Okay, you knew probably some of this because it says stores glycogen, so that we've talked about. We've also talked about how you know, iron can get stored here as ferritin, right? But we also can store other things. So we store vitamins A, D, and B12 here in the liver. So we talked about that. We've talked about red blood cell destruction. So phagocytizes damaged red blood cells and foreign substances and antigens. So we are, you know, engulfing other things as well. We need those Kupfer cells again doing those jobs. Removing toxins, detoxifying. Honestly, that should have been something you learned about. Or you might have learned about in A&P1. I know that I mentioned in A&P1 and Bio 1010, the liver has a lot of smooth endoplasmic reticulum. So when I teach A&P1, when I teach Bio 1010, we talk about the organelles and cells, I always make mention of how there's a lot of smooth endoplasmic reticulum in your hepatocytes, and for that reason, so it's a detoxifier. Okay, and we've talked about this one. The liver makes bile, right? So some of these you probably already knew. You may not know some of these other ones. So let's talk about some of these other ones that maybe you're a little less familiar with. You might be familiar with them. I'm not saying you're not. It's I haven't talked about them. Okay, so oxidizes fatty acids. So fatty acids can enter into the cellular respiration first, or cellular respiration process, but they have to have some things done to them first. So we can oxidize them here in the liver. And, you know, we can also, where is it? Oh, it's right below it. Okay. We can also make some lipids as well. So we can make lipoproteins, we can make phospholipids and cholesterol. And by the way, that's another reason that you need a lot of smooth endoplasmic reticulum here, because the liver makes a lot of lipids. We know that lipid production is performed by the smooth endoplasmic reticulum of your cells. So I'm not going to ask you about smooth endoplasmic reticulum on our test, but I think it is so amazing that you can trace everything, even the big things in your body, to the itty bitty cell. It all goes back to the cell and the organelles inside the cell. So I've always been fascinated by those teeny tiny components. So don't forget about those. Everything can be traced back to the cellular and organelle level. Okay, converts excess carbohydrates and proteins into fat. So you don't have to keep biomolecules all within the same biomolecule type. You can do a little switcheroo, and the liver is good at that. So you can see that there, doing those switcheroos, making some fats. Deaminates amino acids. And hold on, I'm going to sneeze. Excuse me. Okay, pardon me. Deaminates amino acids. So what that means actually is it takes amino acids, which have, we know that amino acids have a carboxylic acid group, an amino group, and carbon chain. Well, deamination means you're pulling the amino group off. And why do you need to do that sometimes? So sometimes you need to do that to send that carbon skeleton into the cellular respiration process. So that's important. And what happens to the amino group? It's going to form ammonia and then form urea. So urea is something that we are going to get rid of some of that in the urine, and we'll mention that again later. But it comes from breaking down proteins. So that actually, this part of this anyway, we talked about in the blood section when we were talking about components of the plasma, right? Because if you have kidney dysfunction, that urea is going to stay in the blood and build up, and your BUN gets high. Okay. Converts some amino acids to other amino acids. So that's, again, the liver is great at doing switcheroos and making things. And here we're not really changing the biomolecule type, but we are going back and forth between one amino acid and another. Acts as a blood reservoir. So we did mention that we have a lot of blood supply to the liver, right? So you can see here it's going to have so much that we consider it a blood reservoir. Okay. Let me erase. Oops. Erase my arrows. And I have a note off to the side there about cut for cells. We did already talk about that. Make sure you know that. Okay, and functions of the liver just in a table form. So you can read that. I'm not going to go through that piece by piece. I think that I will stop this video here and pick up with bile in the next one.